Go ahead, Niklas.
Thank you, Anisha. Okay, so as Henrik said, the Bridge Sweden project. So this is a project that has been going on now for 2 years, I think it is roughly, and it's 1 year or 8-9 months still remaining in this project. And it's the aim is to bridge the gap then between development and production here in Sweden. And just a very brief intro to this is that it involves many different partners or actors in the drug discovery field. So we have me at SweLife Lab at the Drug Discovery and Development platform. We have Stem Cell Center and the pre-GMP facility at Lund University. We have then the different innovation hubs, Stuns and Umeå Biotech Incubator, and then Testa Center in Uppsala. So the idea here is that we have seen mainly from us at the Drug Discovery and Development Center, where we work with academic projects to develop their novel candidate drugs, that the discovery phase we're quite good at, but then when it's time to go into more the commercial and clinical phase, it's a very steep and difficult sort of leap to take for the academic projects. So then this Bridge Sweden project is aiming to sort of make that transition a bit smoother and easier. And one core thing that we sort of identified that is really important in the bridge between discovery and manufacturing and commercial phase is cell line development. So now we have established a commercial cell line development here at the SweLife Lab in Solna. And we are now working with a few pilot projects in developing their cell lines and mainly then take the developed cell line to Testa Center for process development and scale up. And the next phase after that is to then contact a CDMO for actual GMP manufacturing. So I'm really looking forward for today's talk. And I think I'll pause there. You can ask me more questions at the end if there's any question regarding the Bridge project.
Absolutely. And feel free to contact us if you have any questions or inquiries about this Bridge project. Yeah, I'm super happy to have Luisa here. It's like rainy and cold here in Uppsala, so what better way to snuggle up with this cozy seminar. So I'll hand it over to you, Luisa.
Thank you. Let's start to share my presentation here and see if everything works. Can you see my presentation? Yes? Okay. So the main question is why the traditional CDMO model is insufficient for complex biologics. And I want to start with an observation rather than definition. And that is that somewhere between a result that excites a room and the therapy that reliably reaches a patient, something often goes wrong. And it's not on the science, it's in the crossing. That crossing and who should be in the room when it happens, it's what this talk is about. So my name is Luisa Lundin, and I'm the Alliance Manager at NorthX Biologics. Biology doesn't want to be a process, yet every program in the space makes the same journey. You start in the lab, where variability is data, where an expected result is something to explore, where flexibility is how science works. You're heading towards a patient where a therapy has to arrive reliably, reproducibly, every single time. Between these 2 points, it's what I personally call the crossing. Many of you know this as the valley of death, but I refuse to talk about death in innovative therapies. So the crossing has no defined owner, no established process, and no tolerance for improvisation. It is the place where most of the real risk in advanced therapy development actually lives. And critically, it's not only a funding problem, although funding gets consumed here, but it's not a scientific problem. Although the science have to survive it, it is a structural problem. The space has no defined owner, no established process, and no tolerance for improvisation, the improvisation that research environments normally are built around. So what makes this crossing so hard? It starts with something fundamental about biology itself, something the field learned the hard way, and something that actually reframes how we think about manufacturing entirely. The crossing is hard for a specific structural reason. Research environments and GMP manufacturing environments do not just have different rules, they operate on different logics, and those logics are not compatible by definition. In research, variability is data. If something unexpected happens in your bioreactor, that's interesting. That might be even the most important thing you learned this week. But in GMP, variability is a deviation. It has to be investigated, documented, reported, and defended. The same biological event means something completely different depending on which side of the crossing you are on. In research, your process serves your insight. It changes your understanding, how your understanding grows. In GMP, your process is your product, especially in advanced biologics. You do not just make a molecule and put it in a bottle. The living cell that constitutes the therapy are shaped by every parameter, how they are made. If you change the process, you may not have the same therapy in the end. These are not points on the same spectrum. These are different operating systems. And moving a program from one end to another is not automatic. It requires active translation by people that understand it. And if that translation challenge was already significant for conventional biologics, in ATMP, that is the hardest expression of CGT, so cell and gene therapies, this is pushed every single dimension of it's absolutely to the limit. Because in cell and gene therapy, the process is not just important to the product, it is the product. So let's see what it really means. Everything that I have described so far, the two different logics, the translation challenge, the structural gap between research and GMP, applies to biologics broadly. But for advanced therapy, medicinal products, IATMPs, every single dimension of that challenge is pushed to the absolute limit. In conventional biologics, even in the small molecules, a molecule has no identity. It is at least partially separable from how it was made. You can, to some degree, characterize the end product independently of the process. But this doesn't happen in ATMP or cell and gene therapies. That separation collapses. The living cell that constitutes the therapy are not merely shaped by the manufacturing process. They are, in a meaningful sense, its products, which means that if you change the process, you may not have the same therapy in the different package. You may have a different therapy entirely. And this is not a manufacturing challenge to be solved downstream. It is a scientific reality that manufacturing has to be built around from the very beginning. And the complexity does not just add as you scale, it multiplies. Every variable in the system interacts with every other variable. Temperature, dissolved oxygen, passage number, donor characteristics, none of them acts in isolation. So if this is the true nature of the challenge, where do programs actually break down? Because the answer is not where most people expect it to be. It's not in the science. And it's not only primarily about money. We all know that money is important, but it is especially about a specific kind of decision made at specific moments in the wrong environment. So what I mean is that there is a standard narrative about why the programs fail. They run out of money. The value of death is framed as a capital problem, and the solution is to raise more of it. Of course, that you can always pour more money to every process. The question is that these therapies are so expensive that up to a point, it will just run out, or it will be hard even to argue for your funding. So that narrative, in my opinion, is incomplete. And the part it leaves out is the part that matters the most. The programs do not randomly run out of money. They run out of money in the specific moment, and that is the moment of translation, where the translation challenge becomes visible, when process understanding cannot be demonstrated, when the regulatory evidence base is insufficient, when the data needed to justify the next round of investment simply does not exist. The funding gap and the translation gap are not separate phenomena. They are connected, and the translation gap always comes first. When the translation fails late, after all the commitments have been made, after the process has been locked, after the post, depending where you are at in your clinical path, even after the patients are in or near a trial, the consequences are not quite quietly manageable. They are a regulatory notification, a manufacturing hold, a clinical pause, an investor conversation that nobody wants to have. So, what does the decision that determines all of this actually looks like? And where does it get made? Because this is where the story gets specific and where the role of a manufacturing partner either matters enormously or doesn't matter at all. I want to make this concrete because it's easy to discuss process failure in an abstract way that you do not feel its weight. So a process gets locked. It was developed in a research environment, which means it was optimized for insight and flexibility, not for GMP reproducibility. The decision to lock, it felt reasonable at the time because everybody is under pressure to move forward. However, months later, a batch may fail release, and the process that worked in development does not reproduce under GMP conditions. This is not surprising in retrospect. It is catastrophic in practice. Depending where you are at in your clinical path, days after that, a mandatory regular reporting can be triggered before the root cause is even understood, and the clock is ticking in public. Weeks after that, the production stops. The patients who are in the trial or who are waiting to enter it are affected. The program will lose momentum that may never recover from. And when that conversation that nobody in the room wants to have comes, when you have to explain a process failure, a timeline slip, or a certain path forward to the people whose money is funding it. This is not a hypothetical sequence. This is actually the most ordinary consequence of decisions made too late in environments not designed to make them well. I do not have any numbers for biologics, but we all know that in biotech, only 2% of the developed therapies actually reach the commercial phase. This number is far lower when you move into the C and GT field, and therefore this transition is even more important. So, if this is what failure looks like, what is the alternative? The Not the alternative in terms of better luck or to have more capital or stronger science, but the alternative in terms of a genuinely different approach in how this crossing gets made. That starts with understanding what translational intelligence actually is and where it lives. So, what's the alternative? Not better luck. I mean, we're all superstitious, but it comes to a point where you can actually claim that in science. No more capital, a genuinely different approach to how the crossing gets made. We use this term, translational intelligence, to describe something specific. It is the organizational capacity to read a therapy that is developed in the research logic and reconstruct it faithfully in a GMP logic without losing its identity in the crossing. Notice that decision does not say, it doesn't say equipment, it doesn't say infrastructure, it doesn't say regulatory team or QA system or QP. Those things are always necessary, but they are not sufficient. Translational intelligence lives in the people that have spent careers at exactly the cross point between research and GMP manufacturing. And it lives in institutions that have accumulated enough history across enough programs to know which variables matter, which risks are predictable, and where surprises may hide. This last point is very important because pattern recognition of this kind is not transferable in a textbook. It is built through successive generations of increasingly complex biology, recombinant proteins, then monoclonals, then cell therapies. Each one added a layer of understanding that the next one can build upon. You cannot acquire this quickly. You either have it or you're building it. And there are things that this translational intelligence is made in practice. So, pattern recognition. This is very important. Regulatory fluency as a development asset, and proximity to production reality. Proximity to production reality is actually the most practical expression of what I'm describing. It takes a very specific form, a moment in the program life that it's almost always underinvested and almost always the one that determines what is possible later. Let me show you what it looks like in practice. Translational intelligence is a capability, but does it actually get applied? There is a specific moment in every program's life where it matters the most, and it's almost always the most underinvested moment in the entire development journey. When a therapy departs from a research environment heading towards a GMP manufacturing, the most consequential decisions about the future are often unmade. Quality attributes are not fully yet defined. Process parameters are not tested. Analytical strategy not yet built for the regulatory environment it will need to survive. This is the inflection point. It sits between the research origin and the GMP destination. And this place where the decisions that will determine everything get made for better or worse. So the difference between making those decisions well and making them badly is not primarily about talent or intent. It's about the environment. A development environment designed to operate in its proximity to GMP, production reality, is categorically different from another one that it's not. Three things happen at this inflection point when it's handled with the right partner. The first one is co-design. The developer is not just handling work over to a contractor. They are working alongside the scientific team, shaping the process before it's locked. The second one is proximity. The development environment shares walls with the GMP production line. The process learnings transfer in real time and not across a handoff boundary. The third is continuity. Oh, sometimes I stumble upon words, I'm sorry. Continuity. Because the same people that will develop the process remain in contact with the people that will execute them and the GMP. So again, in the field where process and product are inseparable, that continuity is not a convenience. It is one of the most defendable forms of risk mitigation available in the program. I want to show you what this looks like when it works, not in theory, but in a specific program, because the argument for engaging early only becomes credible when you can point out what is actually produced. I want to make this concrete and not in principle in the specific program, as I said. So, this is an example from reality. An allogenic cancer vaccine classified as an ATMP was produced in our facilities for phase 3. The team moved from empty rooms to a first clinical batch in 6 months. The first GMP batch was released within 18 months of the program initiation. And what does this mean? How was this made? I want to be clear what these numbers mean. They are not a story about moving fast and speed. This is not a movie of Fast and Furious. It was not hopping forward. It is a story about making the right decisions earlier while they were still open. And that becomes possible as a result. So 4 things changed because of early engagement. The first, critical quality attributes were defined in a GMP context from the beginning, not in research teams that would need to be translated after, but in terms that you need to be defended at the regulatory submissions. Second, the analytical strategy was built for the regulatory environment, would eventually where it needs to survive. So all the release assays are designed to be both scientifically meaningful and defensible before the process was locked. Third, process parameters were stress tested in the pre-GMP environment, and the edge cases were explored before they became GMP deviations. And fourth, for me, the most important one, the developers were present throughout everything, not arriving at the handoff, not working alongside manufacturing team, working alongside a manufacturing team to every decision point. The last one is not the process detail. It is the thing that made all the other decisions possible. So, if this is what early engagement produces, the question becomes, what does the relationship that makes it possible actually looks like? And what are the honest reasons a developer might hesitate to engage in that way? Before I make any further argument for early partnership, I want to name the reasons a developer might reasonably hesitate. And believe me, in my role as an alliance manager, I've been sitting in this room where all these discussions take place, and I heard the same questions from different developers every single time. These are not straw men. These are really concerns that come up in every conversation we have. we're the people that are considering this type of engagement. The first one is, of course, IP exposure. Most researchers spend too much time and energy with their babies, and of course, they want to protect them as much as they can. But early engagement means sharing, and of course, means sharing the process knowledge and the biological insights, because before everything is protected. This is a real risk. The honest answer to that is that IP conversation is best had early. If you have these concerns, share them, because when the terms are negotiable and the relationship is not yet under pressure, these things can be discussed in a more light environment. And a partner worth engaging will have a clear, documented framework for IP ownership. In most cases, that translates to NDA agreements, but everybody will welcome that conversation and not avoid it. If they avoid it, then that will tell you something important about your prospect partner. The second concern is the loss of process control. Co-designing with a manufacturing partner means that their assumptions and considerations shape your process. Some developers feel, and this is of course reasonable, that this dilutes their scientific authority. However, I want to point out that control and collaboration are not opposites. The developer defines what the therapy needs to be. The manufacturing partner defines what is required to make it reproducibly under GMP. These are complemented authorities. Co-design is not the surrender of control. It's the exercise of it in the right environment with the right partner. The third concern is cost in the wrong moment. Front-loaded engagement has a price at the stage when capital is more constrained. But the honest answer to this is that you need to compare it with the cost of getting it wrong late. A manufacturing hold, a clinical pause, a late-stage process failure. The programs that have to run both calculations arrive to the same conclusion. If you design this first in a good way, then you don't need to do U-turns later on in your process. So if these concerns are real and the answers are honest, what does this actually mean for the decisions that you're making right now or will be making soon? I want to be specific about that, because the question is not really, should I engage a CDMO earlier? The question is, when the decisions that will determine your program's manufacturability are actually being made. Most developers come to the question of manufacturing partnership with one question in mind. And I personally do not know how many times I've heard it, but a lot. And the question is, when do I need to engage a CDMO? It implies that the manufacturing is a downstream step that you reach when you're ready. For me, that question is not wrong, but is the wrong question to lead with. The better question is, when are the decisions that determine my program's manufacturability actually being made? Because those decisions, they are not being made at the regulatory applications. They're not being made when you first call a manufacturer. They are being made right now in your lab, in your process development work, in the conversations that you're having about your therapy with investors and where it needs to be. The question is simply whether someone with manufacturing experience is in that conversation or not. Let me give you 3 specific things worth asking yourself today, not as a checklist, but as a genuine diagnostic. Have your critical quality attributes been defined in the GMP context, or just in the research context, or just in research terms? If the answer is research terms only, that conversation needs to happen now, not when you are filing the IND, so the investigational new drug application, or a CTA, a clinical trial authorization. You need to have that conversation now. The second question is, does your analytical strategy survive the potency assay challenge during GMP? We all know that this is the one we fear, right? So release testing designed for research conditions will not automatically transfer. The gap surfaces late, and it's very painful. The third is, have you had the IP process ownership conversation with a potential partner yet? The earlier that conversation happens, the better the terms and the more honest the relationship is. We all know that in Sweden, we have a huge tradition of collaboration and trusting each other and openly speak about things, maybe a little bit naive, but it is important to be clear and to talk about all your doubts and what you're keeping inside. This manufacturing partner or any type of partner should know all your concerns. So I want to leave you with 3 things that are worth doing differently, not as a checklist, as I said, but as a genuine shift in how manufacturing enters your thinking and when. The first, please map your critical quality attributes now in a GMP context. Do not think in research terms when you are defining and mapping these. You need to have them in terms that will need to be defended at the regulatory submission. So find someone that has seen your type of process through GMP and ask them, which of these will be the hardest to demonstrate control over scale? The answer will probably reshape decisions that you thought they were already made. And the cost of that conversation is very low compared to the cost of not having it. The second, treat your analytical strategy as a program risk, not as a late-stage task. The release testing challenges for, namely ATMP, where you need, for instance, to design these feared potency assays that are both scientifically meaningful and regulatorily defendable, when the starting material, it's inherently variable, are hard. And they don't get easier the longer you wait. So start this work earlier than feels necessary. If your potency assay is not designed for the GMP environment, it will need to survive. You will need to redesign it under pressure. The third, have the manufacturing conversation before you need to, not the procurement conversation, not the capacity booking conversation, but the conversation about the process understanding, about the IP ownership, about what collaboration would actually look like, and have it when it is a strategical choice, not when it's a deadline. A partner worth engaging will welcome that conversation at any stage. The therapies that will define the next generation of medicine are already in development. We all know this. Some will reach the patients. The difference, more often than not, it will not be traced to the science. Let me close with what I think it will be traced to instead. I want to close with reframing this problem simply. The therapies that will define the next generation of medicine are already in development. Some will reach the patients, others will not. And when they fail, the failure will rarely be traced to insufficient science. It will be traced to the crossing, to the decisions made too late, in environments not designed to make them well, by partners who had not seen the problem before. The gap between the laboratory promise and the clinical reality has always existed. What changes is whatever the institute to cross it exists alongside the science, and whether developments engage with it early enough and let it do its work. In the geography of ATMP development and cell and gene therapies development, knowing where that infrastructure lives and what is built from, it's increasingly part of the science itself. I think I will stop there. Thank you, and I'm happy to take questions.
Thank you very much. Yeah, please raise your hand if you have any questions. I will just start off with one. I mean, you talked about this couple of different mistakes or challenges. What would you say is the biggest mistake you see when people come to you at NorthX?
Normally they come later. I don't want to throw anybody under the bus, but even Nicholas here, that is a person that is within this bridge project and has seen so many new therapies in the making. It actually started by saying that the program is a bridge after what it comes. And it should not be seen like that. This interaction needs to be made earlier. You do not want to lock your quality essays. You don't want to lock everything that you defend in a research environment. You want to have this conversation. Is it possible? Is it not possible? Where am I thinking? What am I missing? Did I miss anything? In research, if any reagent is missing from the lab, you can quickly just call and bring another one. You cannot do that in a GMP environment. If, let's say, the natrium chloride that you have defined for your buffer is not available 2 years into the production, you need to go back, start from the beginning, because you have not defined backup products in your plan. And this is reality. And then it's, as I said, your momentum may stop forever and you may not recover from it. So that for me, it's the most difficult or the most common misconception. Yeah.
Thanks. And I mean, like, often when you come to CDMO, you're a big partner, but what else can you help out with? If you meet an early stage project, I mean, of course, the GMP production is one thing, but is there anything more that you can help out with?
Yes. We understood that the importance of early engagement is very important, and therefore we started to be open to engage in alliance and collaboration conversations in earlier than most of the people or other CDMOs, so to speak. We realized that, of course, funding is the problem as well. So we are available to, for instance, help programs to seek funding or financial planning, or have even these pre-discussions sometimes, well, most often without, we've never charged for these. That's what I mean. Maybe I shouldn't say this so openly, but we are open and we are, I think, easy and nice people to talk with. So come and talk to us. You never know.
Great, thanks. Any more questions before we round this up? No? Niklas.
Yeah, I can only sort of emphasize what Luisa said. One thing that we have really learned from this project is to be early to contact the CDMOs and basically to contact everybody, also investors and everybody, to not be afraid to very, very early start talking with people. That's really one of the things that we have taken with ourselves from this project.
And to complement what you say, if you go to an investor and you show that you have thought about how to do this crossing, that you have talked with a manufacturer, that you have, normally this gives another security, it adds another layer of your argument to get funding for your research or for your therapy or your program.
Absolutely. Great. With that said, thank you very much for joining this webinar. We will take a short break during the summer, but we will be back in the fall, and we'll start off with an interesting talk about early stage investments. So that is something that's always the
next part now.
Yeah, the next part. So I hope we will see you in September. We'll send out some information about that shortly. We will also send out the recording of this webinar. So thank you very much. And I hope you had an interesting stay with us here at Bridge.
And please contact me if you have any questions. My email is down there in the slides. Okay. Do I dare to wish everybody a good summer so early?
Yeah.
Yeah.
See you. Bye-bye.
See you.
Bye-bye.
Bye.